Hello, hello. Can you hear me? Can you hear me? Can you hear me? Please, अभी तो चलना चाहिए स्ट्रीम मेरा. Is it working now? Is it working now? I think it should. Right. Okay. Is it start now? Should we start? Should we start? If you can hear me, should we start? Okay. Um, just wait a second. No, just wait a second. मम्मी वे पापा बिस्किट लाए हैं, वे लेके आ जाओ ना. वो अलमारी में रखा है, भाई से पूछ लो. ओके, अब स्टार्ट करते हैं. ठीक है. सो यस्टरडे वी लेफ्ट एट लाइसोसोम एंड आई थिंक तुम्हें सही से समझ में आया होगा कल का लेक्चर. एंड आई ऑल्सो होप दैट तुमने भी कल के लेक्चर के बाद कुछ रिवाइज किया होगा आज, राइट. एंड इफ अगर तुमने नहीं किया तो इट्स वेरी बैड, तुम्हें करना चाहिए. डिड यू डिड और नॉट? क्या तुमने किया था या नहीं किया रिवाइज? यस और नो? ये उस अलमारी में बिस्किट रखा है, ला दो ना. उस अलमारी में बिस्किट रखा है, पापा लाए आज, वे ला दो. अरे तो बिस्किट बाद में खा ले, तो ये बताए. किया था रिवाइज. ओके, सो टुडे वी आर गोइंग टू स्टार्ट फ्रॉम द माइटोकॉन्ड्रिया. ओके. We'll study mitochondria and endosymbiont theory. I mean, it's not so important. इससे कभी क्वेश्चन नहीं आया, but it's a very interesting topic. Okay. And इसके अलावा जो cell surface membrane है, we'll study it. And after that, microtubule organization, we'll study it as well. But alpha tubulin and beta tubulin, it is not so detailed in the NCRT. Also, it is never asked in the IMAT in so much detailed after 2023. Before that, I don't know. Okay. After that, this basic structure like this we'll study. So the main thing is we'll study from this book as well as we'll study from NCERT. Okay. So there will be some topics which we will study from this book. Some topics we will study from the NCERT. It will be totally depending कि किस में जो है किस तरीके से कंटेंट हमको दिया गया है. अगर कोई ऐसा बुक है कि जिसमें बहुत ज़्यादा एक्सेसिव कंटेंट दिया, हम उसको इग्नोर करेंगे. अगर कोई ऐसा बुक है कि जिसमें हमको ये लग रहा है कि नहीं, इसमें ना ये कंटेंट सही मात्रा में दिया गया है, तो फिर हम उस बुक का यूज़ करेंगे. ओके. So now we'll start. Okay, let's. So first, infrastructure of plant cell. We'll study what this is. Well, okay, so today we will start to reach up to this topic. We'll start to finish the ultrastructure of the plant cell. Okay, I think it should be a very good game. Okay, let's start. First, mitochondria. So mitochondria, we all know, powerhouse of the cell. Okay. The structure of the mitochondrion is seen with the electron microscope is visible in figure 1.5. So mitochondria, singular word mitochondrion, they are usually 1 micrometer in the diameter, and they can be very shape. General shape is the sausage shape, or we say cylindrical. They are surrounded by two membranes and envelope. The inner membrane, which forms foldings, and those foldings are called cistrae, which projects into the interior solution, or we say matrix. And the space between the two membrane, this diagram is much better in the NCERT, if I'll show you here. So just let me go there. Yeah, yeah, we can look at this diagram here. So when you look at that, uh, in the mitochondria, it is the outer membrane. This is the outer membrane, and this is the inner membrane. Okay, and here inner membrane is making some sort of finger like projections. Finger like projections, these are called cistrae. Okay, or we say aerobic respiration. Okay, also there's a one property difference between the outer membrane and the inner membrane, which is being that the outer membrane is more permeable as compared to the inner membrane. So if your mind asks the question, how are you defining that the outer membrane is more permeable than the inner membrane? So for that, I'm going to tell you that what, the inner membrane, it needs selective category of things. Okay, and the outer membrane, its property is to transfer the materials to the inner membrane, so that inner membrane can select the materials and it can filter them out for the matrix. Okay, that's why the outer membrane, it will be transferring large amount of substances, which will be essential for the intermembrane space as well as the matrix. Okay, but the internal inner membrane, it will only transfer the those material which will be only helpful for the matrix inside the matrix. Okay, that's why we can justify that the outer membrane is more permeable than the inner membrane. Do you understand that? I hope you do. Okay, now after that we are going to study about also these things as well, that in a mitochondria, there are the site of aerobic respiration. They produce cellular energy in the form of what? In the form of ATP. And the ATP synthesis can occur in very various forms. But why mitochondria is called the powerhouse of a cell? Because it does the what? It does what? It does the aerobic respiration. Okay, and it is the most efficient way of making energy in our human body or in any organism. Okay, hence they are called the powerhouse of the cell. The matrix, it also possesses single circular DNA molecule, and also a few RNA molecules, ribosomes, which are the sentient ribosomes, and the components required for the synthesis of protein. The mitochondria, it divides by the fusion. Okay, um, let let's just let me just go to that topic. Okay? Yeah. So here, the space between the two membranes is called the intermembrane space. The outer membrane consists of the transfer protein called purine. So on the outer membrane of the mitochondria, on the outer membrane there are transfer proteins, which are generally known as purines. There are different type of purines, since a different type of material is just goes inside. Okay, which forms wide aqueous channels, allowing the easy access of small water-soluble molecules from the surrounding cytoplasm into the intermembranous space. Okay, the inner membrane is a far more selective barrier. See, the inner membrane is far more selective barrier and controls precisely what ions and molecules needs to enter into the matrix. The number of mitochondria in a cell is very variable, and it will be totally depending upon the amount of work that particular cell does. Like if you compare a human with a plant, or an animal with a plant, you'll find out that in the cell of a animal, in the single cell of animal, there are more mitochondria as compared to the cell of what, a plant. Okay, why? Because plants, it just stays at a single place. Also, it has a lot of leaves to make its own food, so it do not require too much energy to be made by every single cell. Second thing, lack of locomotion. Plants, they do not do any sort of locomotion, they stays as a single place, whereas animals, they do a lot of locomotion. Okay, they are they are responsible for aerobic respiration. It is not surprising that cells with high demand of energy, right? High demand of energy, like liver cell, muscle cell, will contain large amount of mitochondria. In liver cell, it may contain up to 2000 mitochondria. If you exercise regularly, your muscles will make more mitochondria. Okay, and more mitochondria you have in your muscles, the more strength you have, the more capacity of doing work you have. Okay, function of mitochondria and the role of ATP. As we have seen, the main function of the mitochondria is to carry out aerobic respiration, although they do other functions as well, such as during synthesis of lipid during respiration. A series of reactions takes place in which energy released from energy-rich molecules such as sugar and fats. Most of this energy is transferred to the molecules of ATP. Okay. So what happens that you have studied about the glycolysis. I don't remember it correctly because I haven't revised it, but we all know that what happens that energy-rich molecules such as sugar and fats, they are break down step by step, and the ATP is synthesized during few of those steps. Okay. Mm, ATP is the energy carrying molecule found in all the living beings. It is known as universal energy carriers. Okay. The reaction of respiration, it takes place in the matrix and in the inner membranes and in the inner membrane. The matrix contains enzymes in the solution, including those of the Krebs cycle. Okay, about these enzymes we'll study in the upcoming further chapters. And this supplies the hydrogen and the electrons to the reaction that takes place in the system. So I am not going to explain this to you now because otherwise you will forget it as well. Okay, and most important thing, I don't even remember it right now. Okay, because I haven't revised those chapters till now. So first we'll just do a very basic overview of this topic. Okay, we are not going to be too much deep into that. Why? Because it is purely a database thing, but there will be questions in the IMAT. I think 2025, there were questions from this kind of chapters. Also, there were questions from the muscle functioning. This table kind of questions. This table type of questions where there are different steps and different data in those steps is given. This kind of question came in IMAT 2025. Okay, so we'll take care of these topics later, right? Now we'll focus on the endosymbiotic theory. Okay, so when you look at a mitochondria, it is written that mitochondria has its own circular DNA. Okay, we also see that mitochondria, it also has its own RNA. Sorry, also has its own ribosomes, which are of 70s, right? Even after being in a eukaryotic cell, they are 70s. What does it does? It gives us a suspicion. It gives us a suspicion that mitochondria is used to be a small cell which got engulfed by a large cell, and the endosymbiote theory is just about that, right? And now I'm going to teach you about it. So in 1960s it was discovered that mitochondria and chloroplast, they contains ribosome which are significantly smaller than those in the cytoplasm, right? In the cytoplasm there's ATS, and in the mitochondria and in the chloroplast it is 70s. So they were slightly smaller in the size. Okay, the size of ribosome is measured in the S units, or we say sedimentation units, or we say Swedberg's constant, right? Which are measure of how fast they can sediment in a centrifuge, and also they are indirect measure of what the sedimentation unit is the indirect measure of what. I think you should know this, and you'll have to answer this. Let me just open the comments. You'll have to answer this one. Please answer this. What is the Sedgwick unit is indirect indication of, indirect measure of. Tell me. Sorry, I don't know. No. You have to tell me. Try once. Try anything. Well, कैसे बताऊँ का आंसर मैं अभी देता हूँ आपको. So when you go in the NCERT, um, well, W, um, yeah, here it is, yeah. So when you go in the NCERT, and you go to the ribosome topic, you find here, right, yeah, here, that here S, the Swedberg's unit, or we say the sedimentation coefficient, it is the indirect measure of the density and the size of the ribosome subunits. Do you understand that? Right? You got it? Okay. Yeah. So now we'll start here. The cytoplasmic ribosomes were ATS, while the mitochondria and uh, chloroplast ribosome were SMTs. Okay. It was also discovered in 1960s that mitochondria and chloroplast contain small circular DNA molecules, as I just told you. They also have some amount of RNA as well, also like those found in the bacteria. If you remember that, if you remember prokaryotic cell, in prokaryotic cell, their genomic DNA is also small and circular, and their plasmid DNA, which gives them some additional properties, phenotypic properties. It is also small and circular, right? So it was also found that this type of DNA, which was present in mitochondria and the chloroplast, it was also present in the prokaryotes, right? It was later proved that the mitochondria and the chloroplast are in effect ancient bacteria which now live inside the larger cells, typically of the animal and the plants. See prokaryotic and eukaryotic. Yeah, we'll see that later. This is known as what? Endosymbiont theory. Okay. Endo means inside, symbiont means an organism which lives in a mutually beneficial relationship. How? How does this happens? How does this happens? This happens that the cell, it provides the raw material to the mitochondria, and mitochondria provides energy to the whole cell. Right? It works like that. Okay. Um, yeah. The DNA and the ribosome of mitochondria and chloroplasts, they are still active and responsible for coding and synthesis of certain vital proteins. Okay, but mitochondria and chloroplasts can no longer live independently. So the DNA of the mitochondria and the chloroplasts, it is still in the functioning form. It is not just inactive there. It do the essential functioning of mitochondria and the chloroplast. Also, as we just saw the name endosymbiont, what do you mean by symbiote? In the symbiote relationship, two organisms, they depends upon each other. Okay, so now chloroplast and mitochondria, they cannot live independently. Their relationship with their host cell is sort of what? It is endosymbiotic theory. It is a mutual relationship. Okay. The mitochondrial ribosome, they are just visible as tiny orange dots in the mitochondrial matrix. Um, like if you look at that, these tiny black circles, these are the mitochondrial ribosomes. Okay. Now, cell surface membrane. The cell surface membrane is extremely thin. It is about 7 nanometer in the size. At however, at very high magnification, at least at like 100 times, which we don't give a fuck. Okay, it can be seen to have three layers described as trilaminar. Would you mind if I teach it? Yeah, I'll teach it from the NCRT, because in NCRT it is given in a much better way. Sorry, I'm taking you like from books to books. What do you want me to do? Can you tell this to me? What sort of studies do you want? You want a single book, or you want like content depending upon the different book? Tell this to me. मैं एक सिंगल बुक से ही पढ़ा हूँ, या फिर मुझे ऐसा लगे कि नहीं ये टॉपिक उस बुक में ज़्यादा अच्छा है, या फिर इस बुक में ज़्यादा अच्छा है, ये देखते हुए पढ़ा हूँ, ये बता। हेलो। कहाँ गया तू लेक्चर छोड़ के? Hmm. Okay, so I'll do the lecture tomorrow then. Okay. Bye bye.